Now that we understand how the cell cycle is regulated in cells, let's talk a little bit about, well, what happens when a cell can no longer regulate its cell cycle. Here's our discussion of cancer cells. So we understand that cancer cells don't respond normally to the body's control mechanisms of the cell cycle. Now, how they lose that control, that is still definitely very much unclear. This is one of the reasons why we don't have sometimes very effective treatments and no cures for the various types of cancers, for example, that afflict human beings. This is still an active area of research, primarily because cancer affects so many people and because there's so much more that we still have to learn. We do have some ideas of how maybe cancer cells, some of them might be accumulating the opportunity to stop regulating their cell cycle. And they're listed in this particular slide. So first of all, maybe it's that the cancer cells actually make their own growth factor. They don't have to depend on another cell telling them what to do. If they can make their own growth factor, they can divide whenever and however frequently they choose to. Number two, they might convey a growth factor signal without the presence of a growth factor. This one is a little bit more difficult to understand. So let's say we've got maybe a normal cell, right? And here's my little normal cell, there's its nucleus. And this particular cell has receptors for a growth factor. Let's say here's our little growth factor, it's a little triangle. All right, so anytime the growth factor binds to the receptor that's on the outside of that cell can respond to the signal that that growth factor is sending, which is, hey, you've got to divide now. The growth factor itself, the green triangle, doesn't go into the inside of the cell. Instead, the way that it gets the message across is by changing the shape of that receptor, right? So in the inactive state, or when the growth factor is not bound, the inside tail of that receptor is straight. But as soon as that green triangle binds, then the tail changes shape. And when that tail changes shape, then the cell knows, hey, I'm now activated. I've been given the signal to go divide, and so I'm going to go through cell division. That's under normal conditions, right? The cell will wait for that green triangle, that growth factor, to bind, changes the shape of the receptor, cell then will initiate cell division. Well, what if this particular cell, being a cancer cell, has accumulated mutations? This is one of the ways in which cancer cells appear, right? If they accumulate enough mutations, and those mutations are in genes that regulate the cell cycle, now you've got abnormal genes regulating the cell cycle, and that causes a whole world of problems. And so let's say for this particular cell, the gene that's mutated is actually this receptor gene, and it creates a receptor that always has that active shape on the inside of the cell. So just like what I have drawn right now on the screen, the receptor always looks like that. It doesn't matter whether or not the growth factor is actually bound to the receptor is always in the active state. The cell itself on the inside has no clue what's going on the outside. All it sees is the inside half of the receptor. That one's always active, so it's like, okay, we've got to divide and divide and divide and divide. And it passes on that mutation to its subsequent daughter cells, and those go through division. They divide and divide and divide like crazy, and it doesn't stop. All right, and then lastly, they might have some sort of abnormal cell cycle control system that we haven't figured out yet. So there's still a lot of work to be done in terms of our understanding of cancer. But here's so far what we do know, right? Cancer is a disease, right? When you think of diseases, many of them you think of, well, this is something that I get from outside of my body, right? And when I'm sick, I have a bacterial infection. Well, that's an invading bacterial cell. If I have a viral infection, well, that's an invading virus that's making me sick. The crazy thing about cancer is that these are your own body cells, which have gone through a process of transformation that has taken an otherwise normal cell and through the accumulation of mutations, converted it into a cancerous cell. So it's still your body's own cell, it's just gone rogue, right? Now, the nice thing about your immune system is that it actually has cells that will survey your body and be always on the lookout for these transformed, potentially cancerous cells. The immune system cells are called natural killer cells, or NK cells, and so they look for the transformed cells and they kill them before they have a chance to start really dividing and causing trouble in our bodies. But sometimes, you're stressed or you're ill and your immune system is suppressed, and it might not do that great of a job trying to find these transformed cells. And if the transformed cell is given a chance to start dividing, then it will start dividing, it'll continue dividing until it starts to create tumors. Right? And a tumor is defined sort of as a mass of abnormal cells that's surrounded by otherwise normal tissue. Not all tumors are the same. We have what are called benign tumors, and these are essentially when you have this abnormal mass of cells that only remains at the original site, and it doesn't invade any neighboring tissue, doesn't affect the way that normal tissue functions. Typically, a benign tumor is not something to worry about unless it causes discomfort. Your doctor would probably just say, let's keep an eye on it. It's probably not a big deal. On the other hand, if you have what is called a malignant tumor, malignant tumors will invade surrounding tissues. They disrupt the way that the normal tissue functions, that leads to symptoms. Malignant tumors also have the ability to metastasize. What that means is that parts of the original tumor break off. They travel to other parts of your body and start growing wherever they get stuck, forming additional or secondary tumors. Now, if you are diagnosed with a malignant tumor, for example, and it's localized, it's just in that one spot, you can go through localized treatment, let's say like high energy radiation. And so that particular part of your body will be bombarded with that high energy radiation, and cells that are dividing a lot are more susceptible to damage by high energy radiation. This is because the radiation affects the DNA. And when you're dividing, you are replicating your DNA and you're exposing it, right, to essentially the elements. And so cancer cells, they divide a lot. Their DNA is constantly being exposed. They are more susceptible to that high energy radiation damage than nearby normal cells. And so that will typically damage the cells, shrinking the tumor or helping to get rid of it. Now, metastatic cancers, ones that have spread to other parts of the body or are highly likely to spread, they need to be treated systemically. So you need to treat the whole body. And this is typically where chemotherapy comes into play. So chemotherapeutic drugs are typically anti-mitotics. So they prevent cell division. The trouble is, though, that in your body, you want certain cells to normally continuously replicate, right? Cells like your skin cells. You're constantly losing them, you need more. Or these cells that line your digestive tract, you need more of those because you lose them. Or there are cells in your brain, in certain parts of your brain, that you continuously regenerate, even in adulthood, and you need those to make sure that those are dividing. And when you take a chemotherapeutic drug, it's not specific just for cancer cells. It will prevent all cells from dividing. And this is why chemotherapeutic drugs are really nasty in terms of their side effects, right? People are nauseous all the time because you've messed up your digestive system, right? You can't replenish that lining, and that causes problems for individuals. Or people say that they've got chemo brain, right? They feel kind of foggy. Well, those cells that are replicating in your brain, they're important for things like learning and memory. And if you're not generating those new cells, you're going to have problems in those areas of cognition. So here's just a simple example of how cancer would progress. We're using breast cancer in this case. So you would have, let's say, one cell that goes through this process of transformation. So the cell loses control of its cell cycle, and the immune system does not detect it. And if it doesn't detect it, it starts to grow into a tumor. That tumor grows, gets bigger and bigger, starts to invade neighboring tissue, affects how that tissue functions. Eventually it gets big enough where, it'll reach blood vessels and lymphatic vessels, and some of the cancer cells can break off and use the lymph and the blood vessels to travel to other parts of the body. the cells will get stuck there and they'll continue to proliferate wherever they lodge themselves, and that'll lead to secondary tumors or those metastases in other parts of the body. Now, while we don't have cures and definitely in some cases not very effective treatments, our understanding of cancer and its pathology is advancing every day. The nice thing now is that we've made advances to things like DNA sequencing, where we can, biopsy a tumor and read the gene sequence of that tumor and identify exactly which genes are mutated. And if we can understand that, we'll know, right, whether that particular type of cancer is very aggressive and it needs aggressive treatment options, or maybe this is a slow growing type of cancer that doesn't have the propensity to metastasize, and it can be treated with less severe treatment options. This allows for essentially a more personalized treatment for each cancer patient, which increases their prognosis and sort of decreases the amount of suffering that they have to go through. If they don't have a very advanced form or very aggressive form of cancer, they don't have to go through the chemotherapy and all the really nasty side effects that goes along with it, and they can still go into remission and do very well. All right, the last thing we have to talk about now is sort of a divergence. We're going to talk actually about cell division in prokaryotes. So far in chapter 12, we spent all this time talking about the complex process of mitosis, which divides eukaryotic cells, but prokaryotic cells need to divide as well, right? For prokaryotic cells, this process of cell division is called binary fission. And remember, because prokaryotic cells like bacteria and archaea, they're single-celled organisms. If you divide a cell, you're really going through asexual reproduction to make a new organism. And lucky for us, binary fission, again, is simple, and there's not nearly as many processes as what you have in mitosis. It's literally 3 steps. So the first thing the cell needs to do, just like eukaryotic cells, is to replicate its chromosome. Then this happens, it starts at what is called the origin of replication. So this is essentially just a sequence of the DNA nucleotides that says, start replicating me here, and the cell will start. As the chromosomes are being replicated, they actively move apart to opposite poles of the cell. And once the chromosome is finished being replicated and has moved to opposite poles, then the plasma membrane is just going to pinch inward and divides the cell into 2. And so this is kind of what it looks like. So we've got our E. coli cell here, a type of bacterial cell. Here's its circular chromosome in blue. You can see in green here the little segment, that's the origin of replication. This is where the 2 DNA strands are pried apart, and each one is then used as a template to replicate the chromosome. The 2 copies of the chromosome, now each with their own origins of replication, are going to move again to opposite poles of the cell. And then once the chromosome has completed being replicated, then the cell will essentially just pinch in half, and you've got 2 daughter cells that are, again, genetically identical to the original parent cell. They're clones of each other and the parent.